Hello everyone, I'm Nicolas Brich from the Institute for Bioinformatics and Medical Informatics IBMI at the University of Tübingen. Today I will present our paper titled visMOP, a visual analytics approach for multi-omics pathways. First, several concepts have to be explained. The first one being, what actually are omics? Omics is a suffix denoting different disciplines of biomolecule measurement. This is best illustrated using the most frequently characterized ohms, which are the targets of the omics. Transcriptomics, in which the transcriptome, the collection of all transcripts in a sample, is measured. Proteomics, where analogous to transcriptomics, the proteins are measured. And metabolomics, in which the metabolites, which means most small molecules, are measured. The second concept is the one of biological pathways. These are maps of the synthesis and catabolism, meaning the utilization of biomolecules, which essentially describe how an organism works on a molecular level. Most biochemists or biologists will have encountered the visualization on the right, published by Roche. In essence, it is a large directed graph of biochemical reactions. It was first published in 1965 and thus is a reference for generations of researchers. It's analog and thus offers limited informatics analysis potential. Databases of digital pathways exists, however. A very prominent representative of this is the KEGG database, the Kyoto Encyclopedia of Genes and Genomes. Despite its name, it's not limited to those, however. It was released in 1995 by the Kanehisha lab. KEGG contains biological pathway diagrams as networks, similar to Roche, in which molecular entities and their reactions are shown as directed graphs. They are organized in a rather flat hierarchy, with connections between them but not so strong hierarchical organization. KEGG is widely used, offering quite a lot of utility, like a REST API and web interfaces to query specific entities. A more recent development is the Reactome database, which is part of the Elixir organization, which is a pan-European, partially publicly funded organization bundling bioinformatics resources under open access. Reactome is, in contrast to KEGG, a more hierarchically structured pathway database. Reactome consists of a general overview and an associated detailed pathways. The very coarse topics are assigned to the roots of a hierarchy, corresponding to an overview detailed pathway. Here, other hierarchical topics are arranged in a small graph. More fine-grained topics, reaching down to the level of single reactions, are arranged in the lower hierarchical levels, which resemble the pathway maps in KEGG. With the most important background knowledge out of the way, I now want to outline the starting point of visMOP, pointing out our aims and the challenges we faced at the beginning of the project. Our cooperation partners from the laboratory of the Universitätsklinik Tübingen use a set of distinct programs for each of the omics analysis. This makes integration of results difficult. Our main objective was thus to integrate those multiple omic analyses for visualization and visual analysis. We think that for a given experiment, this would result in a more representative picture for the actual processes in question. We also want to use known biological pathway representations to make use of existing mental maps and previous knowledge. One conflict with this is that existing resources like KEGG or Reactome do not take the supplied data into account when generating a layout for the pathways. We, on the other hand, want the supplied data to influence the layout of the visualization. Primarily, visMOP should thus constitute an entry point for further data analysis and hypothesis generation by supplying an overview over the data. With this, I want to give a brief overview over the application itself. Here, as you can see, the central element of the visualization is a clustered graph. Each of the nodes corresponds to a metabolic pathway as defined in Reactome, visualized with a circular glyph. Each cluster constitutes a group of similar regulated pathways, while the light area are the unclustered pathways. On the outside, the Reactome hierarchy roots are displayed. We call them superpathways. There are various settings to influence the main visualization, which can be seen on the left side, and I explain them further on. As the clustered graph only uses the pathway identity in accordance with previous knowledge, that is, existing databases, we, on demand, show a detailed pathway in the Reactome style. But how is the input data, meaning experimental omics data, transformed into the visualization? And what is omics data anyway? Most of the time, this will be a fold change. A fold change indicates a fold level difference between two measurements, most commonly a control versus an experimental condition. And this is on an entity level, meaning per protein, per gene, per molecule. This can be in the hundreds of measurements for metabolites to 10,000s of measurements for transcripts of genes. In our pipeline, we begin by first selecting our omics data sets and optionally applying filtering to it. The data is then automatically mapped to reactome pathways. We then derive variables describing the pathways based on the supplied data. For example, this can be the percentage of measurements of pathways for each of the omic types falling in or outside of a specific value range. These derived values are then used for clustering. One problem with clustering in higher dimensions, however, is that the space becomes more sparse the more dimensions are added. This is known as the curse of dimensionality, and this makes clustering less practicable. We thus utilize dimensionality reduction in the form of UMAP to bring the representations into a lower dimensional space. We do this twice, one time for clustering, in this case n divided by 2 dimensions, and one time for the visualization, for which we target a 2-dimensional space. We then use optics to cluster in the lower dimensional space and assign the nodes in the 2-dimensional space to respective clusters and calculate centroids for each cluster. The resulting data now constitutes our visualization data. Using the cluster centroids and the amounts of nodes for each respective cluster, we generate a Voronoi tree map. Within those clusters, not the original UMAP position is used to place the nodes, and instead we use a force layout to group the nodes according to their superpathways. Those superpathways are mentioned earlier, arranged in a circle around the tree map. As the nodes and the glyphs which are used to represent them are a central element, we now more closely investigate them. The glyph is a radial pie or donut style glyph with a segment for each omic. The amount of segment scales globally with the amount of supplied omics, meaning if we only input 2 omics data types instead of thirds, we would get semicircles. Radially, the glyph is segmented into 2 rings. The colored swatches on the outer ring correspond to individual entities like proteins. They are colored on a blue to white to red diverging color scale, depending on their associated measurement value. The inner ring indicates how many entities of a given type, for example proteins, are present in the pathway and how many of them were actually measured in the given experimental data set. The glyph exists in 3 detail levels. In the highest detail level, used when zoomed in closely, the glyph appears as shown previously. When zooming outwards, a low detail glyph is drawn instead. Here, the averages over each omic type are used to color the segment. Zooming outwards even further causes an aggregation of these per pathway glyphs into per cluster glyph, otherwise using the same style as the intermediate zoom level. The super pathway glyphs are drawn following the same rules, but without having a per cluster stage. These glyphs are also used in the pathway glyph comparison functionality of the tool. This is an interactive side-by-side view for multiple pathways. The order of the cards can be changed via drag and drop. Here, the high detail version is used, and additionally, the glyphs become interactive, allowing to get numerical values for quantities like the individual measurements, as well as the averages for the omic types. The overview can be used to reveal general patterns across the clusters. For example, the cluster outlined in red is the only cluster having an average upregulation across all omics. Orange and teal show upregulation in transcriptomics and downregulation in proteomics. They differ, however, in that orange does not have any metabolic measurements mapped to the contained pathway, which is highlighted in the orange-teal dashed line. As you all probably know, a graph is defined as a set of vertices and edges. Up until now, none of the edges were actually shown. The edges in our clustered graph indicate hierarchical and logical connections. Hierarchical edges indicate connections in the reactome hierarchy. Logical connections might be there if a product of one pathway gets used in another pathway which is not part of the current hierarchy. When showing all these edges, however, the result is a cluttered graph, as seen on the left. We thus, by default, do not show any edges. Hovering or selecting the node, however, then reveals the connections for the hovered node. While we believe that the overview visualization is already a very helpful tool, some investigations need access to a more detailed level. We thus add another view, a detail view. As stated in the beginning, we utilize the reactome pathway layouts and symbol encoding, which is related to the SBGN, the Standardized Systems Biology Graphical Notation. Each molecular entity type is mapped to a specific glyph, like proteins having rounded corners and small molecules being ellipses. We color the entities to the color mapped to the corresponding measurement value. As some elements map to multiple classes of measurements, for example to proteins and genes, the elements are subdivided. Other elements, called complexes, are a combination of several types and multiple entities of each type, acting in concert to partake in biological processes. Thus, to properly display them, they are subdivided in thirds and colored by average. To see details concerning the composition, they can then be clicked, displaying a high detail glyph with interaction capabilities. To investigate ideas for possible hypotheses, the user can also interact with the input data in tabular form. The table is searchable and sortable. Selecting entities results in highlights in the main graph, indicating setwise unions and intersections in blue and green respectively. Vice versa, selecting a pathway in the main overview highlights all contained entities in green in the table. In addition, there are a set of filters helping to hone in on interesting data points. These filters include inclusion and exclusion filters based on the superpathway, meaning the users can easily show only the pathways which are contained in a specific pathway or explicitly hide those. And numerical filters based on the number of measurements per omic or in total. Here users can show pathways lying either in a specific value window or flip the filter hiding all the entries in the specific window. Finally, I will showcase an example workflow based on data from a study published previously by the co-authors. In the study, the effect of a high-caloric diet on the lipid metabolism was investigated. As the study was concerned with the effect on the metabolism, we can first investigate pathways related to metabolism. Here, this is shown by using filters. One, to filter out pathways not containing any metabolite measurements, and interaction to highlight the connections. Alternatively, an additional filter could be set based on inclusion in the metabolism superpathway. Here, our visualization reveals two interesting clusters with high upregulation across all omics. To further investigate this, we then can use the detail view. The glyph and the detail view of the metabolism pathway show that the general trend of upregulation is visible there as well. This holds especially true for the metabolism of lipids subpathway, which is highly important concerning the topic of the data. This can again be seen when selecting the pathways in those clusters belonging to the metabolism of lipids superpathway. This is in agreement with the original study, as a fat-rich diet causes a need for increased lipid metabolism, while on the other hand, a reduced lipid synthesis due to the abundance of lipids in the food, as seen for example here in the fatty acyl CoA biosynthesis pathway. To summarize and conclude, we present visMOP, a visual analytics framework for multi-omics pathways. The visualization features pathway-level glyphs with a data-driven layout to offer an exploratory analysis workflow. The overview abstracts the data while using underlying structures to aid the exploratory analysis, while the detail visualization allows for the development of a more focused hypothesis for downstream analysis. We then enrich the visualization with interaction to increase the visualization's analysis capabilities. With this, I would thank you for the attention. If you want to know more about visMOP, please check out our paper.